Yes.
Slides up somewhere?
So these slides are on that GitHub page. You should be able to reach it over the Wi-Fi. If you can't reach it, it's because your company is blocking that site. Anybody's getting blocked from that GitHub site? github.com/May Institute. Saw a couple yes. Are most people able to get it? You or get it? If you would like, I can try to send it to you guys right now. I just sent the slides on email too, if you're being blocked from the GitHub site.
Thank you.
Okay, let's go ahead and get started with day 2. It looks like most of you are here for the Sky Jam, so hopefully that was a nice review of everything we learned yesterday about the target list, about retention time, about kind of getting familiar with Skyline for setting up the method. And today we're going to take the values that you would get from that experiment and make them worth something, essentially. Today we're talking about absolute quantification and method validation. This morning, right now, I'm just trying to give you an overview of all the things we're going to launch into much more depth in today. So if something goes by really fast, don't worry about it. We're probably going to be returning to it later today. So what I want you to walk away with from this 1 hour presentation is that there's a lot of validation criteria out there and a lot of different kind of opinions and views on what validation is and what validations are necessary. So I want you to not necessarily get stuck in your mind that, oh, this is like a checklist of validations I have to do, but kind of more, what validations does my experiment specifically require? And I want you to think about how you can prioritize those validations so that you don't end up wasting a ton of time on targets, on experiments that are essentially going to be doomed to get cut out of your assay anyway. So we're going to start with absolute quantification. So yesterday you learned all about setting up a targeted MS method. We talked about all of these different aspects of a targeted MS method, both from the mass spectrometer side of things. Yesterday morning we talked about all of that. And then from the liquid chromatography sides of things. So you've built this targeted method and you're getting all familiar with Skyline, but now what? So now you've measured these peak areas, and let's say, for example, you just have a very simple experiment where you have maybe a healthy and a diseased sample, a a control and a mutant sample, and you get these two peak areas, and I've been really nice to you, and in Skyline I've normalized your y-axes, so you can just tell visually that the sample on the left has a much bigger peak than the sample on the right. But does it? And this is where we get into validations. How can we turn these peak areas into an absolute abundance? Because when you think about quantitative mass spectrometry, the quantitative part doesn't come from the mass spectrometer. There's nothing inherently quantitative about a mass spectrometer. It's the tricks and the experiments and the statistics that we can apply to these essentially unitless peak areas that make it quantitative. How do we do that? We calibrate and validate. Those are the two buzzwords of the day. Please get those burned in your mind. Calibrate and validate. It's not enough to just have a peak area. If you want to do absolute quantification, for relative it's fine, but for absolute we have to go farther.
So
yes.
The peak areas aren't actually unitless. They are estimations of the number of ions delivered in that range. It's just that the number of ions delivered is so impacted by all kinds of different,
They're unitless in terms of units that most biomedical and biological research would care about, like protein copies or number of tumor cells or femtomoles of disease. All of that is something that we have to apply to the peak area. The peak area itself doesn't tell us anything besides, like Brendan said, just the straight number of ions that went into your detector, went into your Orbitrap. So Sue showed you yesterday this image that you're probably familiar with precision and accuracy. So what we want to do, kind of like with any other calibration, when we calibrate this, we want to make sure our input amount, our known input on the x-axis, scales exactly to a measured output from our mass spectrometer. So we want to take the peak areas and relate them through some calibration metric to a known fixed amount of input that we're confident. So your known quantity, and this can be maybe a standard that you spike in of the protein level or of the peptide level, should be able to be converted to a measured signal on your mass spectrometer. And a lot of the validation criteria you're already familiar with. So one of them is that this should be reproducible. If I measure the same known quantity over and over again, I should get the same measured signal from my mass spectrometer. That peak area shouldn't change if I know I'm putting the exact same known quantity into it. This should be linear. The signal should be linear. And Sue talked yesterday, introduced you to the concept of linearity, and today we're getting much more in depth into the concept of linearity. But this known quantity should scale 1-to-1 to a measured signal. And unknown peak areas can be converted into an absolute quantitation value using Skyline. And Sue will be showing you hands-on how you can do this, where you can have a single point, a 1-point calibration, where maybe you just have one set known amount that you've measured the peak area for, or you could have multi-point, which is of course better, multi-point calibration, where you can gain more confidence in this regression line, so that when you measure an unknown peak area from your mass spectrometer, so here we're showing light and heavy peak area ratios because this was a gold star A+ quantitative experiment where we had standards. This red arrow is indicating this is on the y-axis the peak area that I got. So now that I have this regression line, I can follow that down to the point on the x-axis, which is my absolute quant. So I can take my peak area on the y and convert it down to an absolute quant on the x. So these measurements should be reproducible. They should be linear. We should validate them. But in a perfect world, this is not always a straight line. So Sue introduced you to some of these calibration curves where she showed you maybe on the top end at the high amounts of known quantity, so on the far right side of this known quantity axis, you can start to get a plateau at the top. And we talked a little bit about saturation, that was indicative of saturating the mass spectrometer, saturating the detector. But actually, we can get the tailing on the low end too. So calibration curves, when we take a number of known quantities, samples with known quantity, and measure their peak area, a lot of times we'll get kind of a flat line on the end, a hockey stick shape. And this is because we are floored, our signal is floored by the noise in the mass spectrometer. So Skyline subtracts out noise from the peak areas that it reports. So a lot of times when you look at the chromatogram, it goes straight to 0 on the y-axis, right? And that's because Skyline has detected what the noise level is and then subtracted it out. I see a little bit of confused looks. Do we want to go a little bit more into that?
Yeah, I'm confused.
So the noise level means that, and I can show you some examples. What it practically means to you is that there is a limit of detection for what you can measure in a mass spectrometer. And this is true for every analytical instrument, not just mass spectrometers, right? So you've probably heard maybe of LOD and LOQ. If you're from an analytical chemistry background, if you're from a biological background, maybe this is new to you. Buddy up with an analytical chemist in the class, and together you'll have everything covered. The limit of detection means there is a fixed known quantity below which it doesn't matter, I can't detect it. I can't say, for example, if this is the number 10, if there are 9, if there are 8, I can't detect anything below 10 molecules with my mass spectrometer. 10 proteins, let's say. If there's 10 copies of the protein in my sample, I can't detect below 10. The limit of detection and the limit of quantitation are slightly offset. So if you detect something, it doesn't always mean you can accurately quantify it. That's a kind of really important concept, that you can't quantify everything you detect. And I see nodding heads, so it looks like everyone's okay with just accepting this for now. Later today, Cyril, who's here at Northeastern, in Olga's lab, is going to talk to you about his method for calculating LODs and LOQs. And D. Armani will be in here to talk about some more methods. There's a lot of different methods, but the big take home is that you cannot quantify everything you detect. So calculating this limit of quantitation is extremely important, because you want to be able to say, if I get a number from my mass spectrometer in my absolute quantification, if I get a number below this threshold, I shouldn't trust it. All I can say is that relatively, it's different. Okay, we'll be going way more into depth with this. I think we have an entire afternoon dedicated to this concept, but I wanted to introduce you to it so it's not totally out of left field. These experiments are called a calibration curve, and we'll be going, there's a whole section on it this afternoon. So absolute quantification, absolute quantitation also requires other method validations, and why is that? So most of you are going on to do some more statistics, and I just want to emphasize that you can have the perfect statistical analysis, but if you have garbage data, the best statistics in the world won't rescue your experiment, because you'll still get garbage results. You can have perfect data, but then if you have a garbage analysis, you're still going to get garbage results. So it sounds like a lot of you are going to be here learning more about R, learning more about statistics for reproducible statistical analysis. So I just wanted to impress upon you that even the best statistics in the world, even the best batch effect normalization and the best machine learning models cannot rescue bad data. And there's a lot of different opinions in the field for method validation. So in mass spectrometry, in the community, the mass spectrometry community, this kind of started in about a decade ago, in 2003, with this establishing the fitness for purpose paper. And that spun off a couple other papers, because one of the points they were emphasizing in this literature was that any uncertainty in your method should be measured and documented. So any source of variation, any source of uncertainty in the quantities that you're reporting, you should already know about. They shouldn't be hand waved away. We should accurately define them and characterize them. And so a lot of the other kind of big players in the mass spec proteomics field started writing their own thoughts on validation of mass spectrometry methods. So there was a paper from Russell Grant and Andy Hufnagel, so they do a lot of clinical proteomics work, that described their opinions for what kind of validations they would like to see the community putting out for clinical proteomics. A whole host of leading proteomicists, mass spectrometry proteomicists, put out another paper for the best practices using a fit for purpose approach. And in that latter opinion piece, they talked about different tiers of assay validation, where the top tier would be things like clinical mass spec proteomics that should have absolutely no uncertainty left unturned. Like every uncertainty should be eliminated, and what we're measuring should be absolutely characterized. And that included all kinds of tests and validation experiments that you should do. But they also acknowledged that not every mass spec proteomics experiment was going to be a clinical assay being used on human subjects. So they also pointed out that tier 2 and tier 3, which might be more exploratory or more research grade mass spec proteomics, doesn't necessarily require all the same validations, or maybe any validations if we're just looking to see what we can see. And these papers go into great depth explaining what these experiments are and the exact tests and what they're testing for, and what how we should interpret the results. So if you wanted to get more into the details of these, I encourage you to read these two papers. Neither of them are particularly long, and they're not particularly difficult reads, and they really summarize the various opinions in the field. And all of these experiments, we'll be going into depth later, and I have a real data set that was published from Andy Hufnagel's lab that shows all of these types of experiments, and in Skyline you can play with the data, and we'll kind of get more familiar with what the data looks like and how we can analyze it. So I wanted to make a note that now that DIA, data independent acquisition, is becoming a popular method in the mass spec proteomics field, you are not excluded from having to do these calibrate and validate experiments. If you're not familiar with data independent acquisition, sometimes also called SWATH, I call it DIA, I wanted to give you a quick analogy of what this is. So imagine you're in a library and there's a ton of books everywhere. If we were doing targeted, we could go into the library and just pick out our books and put them on a cart. So now we know what books are on our cart. We know that, for example, maybe we picked out Moby Dick, maybe we picked out War and Peace. We know the books that are on our cart. But now let's say we go back to our cart and we say, is Harry Potter in our library? Well, if we just have our cart, we couldn't ask that. We would have to go back into the library and then search around and grab Harry Potter and put him on our cart. With DIA, you get the entire library. And just looking at the entire library can be very difficult to tell what books are in your library. So we know, though, all the titles of the book. So if we know the title is Harry Potter, we can go into that library and say, okay, A, B, C, D, H. H is where Harry Potter should be. You can scan the shelf of H and say, oh, here's Harry Potter. So DIA lets you acquire the data and then go back and ask however many questions you want. Is this in my data? Is this in my data? Is this in my data? You can pull out a cart's worth of peptides or proteins from DIA data and just ask, here are the things that I said I cared about. Or you could take that cart and put it aside and then pull out a whole different cart and do targeted analysis of DIA that way. You are not excluded from calibrating and validating if you do this with DIA. So if you wanted to measure an entire genome, if you wanted an entire proteome profile using DIA, could you target every protein using SRM, using PRM? You could argue yes, with enough SRM and PRM methods. If you use 100 different methods to target however many 10 10,000, is it 10,000 proteins in the human genome? I think there's spliceoforms too. But you would have to compromise if you use PRM, right? Something like PRM or SRM wouldn't necessarily be comprehensive. It's targeted. You would just be asking a set number of questions. But if you use DDA, maybe you would be more comprehensive, but it's certainly not great quantitation. So DIA, you're sacrificing a little bit of the comprehensiveness, and you're sacrificing a little bit of the quantitation quality, but you're getting a little bit of both. So on the left, yes?
Can you go into a little bit more detail about how you're compromising the comprehensiveness? Because according to your Harry Potter example, it seems pretty comprehensive.
So it kind of a little bit, to tie it back to the limit of detection we were just talking about, with DDA, you can dig a little deeper into a sample than you can with DIA, because DIA starts to get a little bit noisy. The data gets a little noisy with DIA. So it's kind of an active area of research. How good of comprehension can we get with DIA versus DDA? But there's compromises with DDA too. The way DDA works, it's stochastically picking the tallest peaks, right? So if you do that 3 times on the same sample, you get slightly different results each time, which is why targeted methods like SRM and PRM are great, because you know exactly what you're getting every time. As far as the workflow for comparing this to what we were learning about all day yesterday with SRM, you start with the hypothesis generation, but DIA, you start with the data acquisition. You don't have to know too much about your sample when you start with DIA. You ask the questions after you've acquired the DIA. The workflow for generating the mass spectrometry method, which are the blue boxes, is also a little bit different. With DIA, we don't necessarily need to prioritize the retention time scheduling quite so much. You need to have good chromatography, but it's a little bit different. So I'm going to put this slide here, but I'm not going to talk about it too much because there's a great paper from Jared Eggertson, who's now in Boston, and he wrote all about like this huge protocols paper with every detail you could possibly imagine if you wanted to try something like DIA for quantitation. And most of the analysis for DIA is done the exact same way you were learning yesterday, where we would put targets into the target pane in Skyline, and then we would just import our DIA data, import results, import the DIA file. But it's extremely messy. DIA data is very noisy, so the analysis can be a little bit of a mess. So as far as peak picking, it can be very important to have retention times. Just like we did yesterday, knowing when the peptide is eluting in the chromatography is very important to help us pick what the peak is, because it's a mess. It's a mess if you had to do this without retention times. I don't know how you would know where to look. It is still not inherently quantitative, though. So just because it's DIA doesn't mean it's any more quantitative. In fact, it's a little bit less accurate quantification than something like SRM or PRM. So we have to calibrate and validate. Even if you're doing even if you're doing something like DIA, you should be calibrating and validating your quantification. So Sue will be going over in great detail how to make a calibration curve for an SRM or a PRM, but I just really wanted to emphasize, because this is kind of my, all of my attention the past few years have been on how do we calibrate and validate at scale. So how can we make a calibration curve for a targeted mass spectrometry experiment where we have tens to maybe hundreds of peptides, so the type of stuff that we were learning about yesterday. And typically, you synthesize. You synthesize and use the light-heavy ratio of your endogenous peptides to your synthesized heavy isotope standard. But with DIA, you're usually not doing something on the scale of tens to hundreds, because if you were, you would just do an SRM or a PRM, depending on your instrumentation. For DIA, you're usually interested in thousands to tens of thousands of peptides. So it becomes a little cost prohibitive to synthesize that many standards. So the bulk of my research for the past couple of years has been how can we make a curve without synthesizing that many standards. So what we've been doing, and together with Andy Hufnagel, we've been playing around researching the idea of using a background proteome to make a calibration curve. So I'm not going to go too into detail on this, and all of this is unpublished, so don't tell your friends. I'm not going to go into too much detail because this is a targeted course, but the point is I want to very vigorously emphasize to you, DIA does not excuse you from having to calibrate and validate, and there are ways to do it, so no excuse. What you measure in a calibration curve for something like SRM would usually be the ratio between your endogenous and your synthesized heavy. If we're not synthesizing, what we have to use, all we're left with is the label-free peak area. So this is what I mean by it's a compromise with DIA. It's not as vigorously quantitative, as rigorously quantitative as something like SRM or PRM. But that said, you can still come up with a calibration curve using this idea that we're encouraging called a background proteome. And the background proteome can really be anything as long as it's matched to the proteome you're looking at. So, for example, by matched, I mean the chromatography has to be very similar. So we saw yesterday how things like gradient and column can change the chromatography, but the matrix changes the chromatography too. If you have a less complex matrix, your chromatography will shift compared to a very, very busy, complex matrix. So if I spike a heavy peptide into something like depleted plasma, it will look different than something like neat plasma. I've removed a whole bunch of the complexity from the plasma. Neat being, I just took it straight out of someone's arm, and depleted being, I took away like the top 10 most abundant proteins. Something like if I'm comparing yeast and spiking a peptide into yeast versus if I spike it into human. The complexity of the proteome changes the chromatographic properties. The other thing that's important to match is the mass to charge of your background proteome. So for the most part, peptides that we look for are maybe 400 to 1,000 MZ, whether that's doubly or triply or quadruply charged peptides. So not to linger too long on this, but take home some examples could be, say, human plasma and chicken plasma. So Andy Hufnagel is working on doing this kind of thing for human plasma, and he's going to dilute his human plasma into chicken plasma. Should have very similar chromatographic properties, and as long as he excludes any shared peptides between the two species, any conserved proteins and peptides between the two species, that is an example of a background and a proteome of interest. So maybe you have a human cell lysate, maybe you could use something that is sufficiently diverged from human, but another cell lysate.
So how do you want to find the background proteome?
I am not. I am just using it as a diluent. I'm not using the background proteome for anything except for a diluent. If I just used water in human plasma, and I just kept diluting my human plasma with water, that changes the chromatographic properties, and it also changes the peak area. It changes how we measure it. It actually makes it a little bit more sensitive if we keep diluting it because we're making it a simpler mixture. So we need to replace our proteome of interest with something equally complex, but that won't interfere with the signal from what we're actually interested in. And what this ends up looking like, and Sue will be showing you a lot more examples of calibration curves, but just to show you in this kind of label-free kind of way, if we don't dilute at all, we should get a nice tall peak. If we dilute it by half, we should expect, linearity-wise, that the peak should decrease by about half. And then if we reduce it tenfold, we should get a peak that is about tenfold smaller. So in these whole proteome calibration curves, that's exactly what we see. But one of the advantages of doing this, at least with DIA, is that we can do thousands to tens of thousands of peptides. We're not limited to 10 to hundreds of peptides. And it ends up looking exactly like you should expect, where the majority of the signal is in your undiluted point. This is just a heat map where each row is a peptide, and I have 15,000 peptides in here from the yeast proteome. We get the most signal out of our undiluted point, and then we see the signal fade down as we dilute out the proteome of interest. Just to flip this figure on its side, so now you're looking down in a box plot, it gives the canonical hockey stick shape that we would expect to see. You notice some peptides don't look super great. They're not super quantitative. That doesn't mean anything's wrong with the method. It just means those aren't great quantitative targets. So DIA is also not excluded from any of the validations. So now we've shown how we can maybe do a calibration curve, but you're not excluded from validating too, which is very easy. For reproducibility, the field likes to use something called the %CV. And Sue will be showing you how to do that, and you'll be getting a lot of hands-on for how to do that in Skyline. But the short story is you're shooting for under 20% CV. And here I'm showing you a CDF, a cumulative density, and 20 is here on the left, and this just means that the majority of peptides are under 20. So I get about 13,000 peptides are very reproducible with this method. 13,000 peptides approximately have an under 20% CV. So I saw a question in the back.
Yeah, so since you don't really have any confirmation here with your heavy labeled peptide, how many transitions you would consider as a minimum number for the positive ion? Because of course now you have 10,000 peptides.
Yeah.
So how you make sure that this is the real correct one?
So the question's about how do I know the peptide is the correct one? How many transitions would I use to establish that it's the right peptide that I think it is? So the short answer is, and I'm not sure if Sue will actually be going over this, 3 to 5 transitions tends to be the rule of thumb. Would you go higher than 3 to 5? Or is that like a pretty decent floor?
Well, in the triple point world, you would need a minimum of 3 to help ensure identification, and that's helped if you have internal standards. If you don't, to be on the safe side, I would go with more. But with DIA, since you're selecting them all, the bigger question is how many do you use to quantify? And so right now, I don't believe there's consensus in the field because, as far as I understand, most vendor software packages allow you to select one for quantitation and the rest are confirming. Or if you decide you want to use all of them for quantitation, it does them individually. So it doesn't group them together because, again, I don't think there's yet in place a thoroughly vetted software package that can ensure that there's absolutely no interference and that you will get false positives if you group all the transitions together.
So just to summarize, hopefully I could summarize for those that are listening at home. Sue recommended 3 to 5 was okay for something like a triple quad when you're doing SRM. For DIA, there's not a great hard and fast rule yet because it's still kind of Wild West with quantitation and DIA, which is why I am emphasizing you are not excused with DIA from doing calibration and validation, even though it is the Wild West. It's good hygiene.
I'm also going to say that statistically, every time I've looked at it, Mike McCoss, my boss, is very adamant that you should use all transitions, and even in low selectivity environments like using MS1 chromatograms, I have always, and so like the lower the selectivity, the higher the probability that you have interference, right? So the less selective your experiment is, the higher the probability that you have interference. And I've heard people like do MS1 quantification where it's much lower selectivity than either DIA or SRM say, oh, you should only use the monoisotopic peak. But every time I've gone to check it out statistically on very large sets of data, using a single peak statistically, on average, ends up being worse than using all your collective things. So take that for what it's worth. I mean, if you're doing a high precision experiment and you can bet everything, you may choose transitions that you know don't have interference. But if you're just statistically on average want the best quantification, I will stand by Mike's assertion that yep, you should just be totaling all the transitions. And that's kind of what's behind it.
To summarize, Brendan, extremely briefly again, just like I did with Sue, Mike McCoss, hi Mike, would recommend everything, all the transitions, all the transitions. For me, for doing these DIA, I do 3 to 5, and I have a method for interference detection, and there's a couple Skyline ecosystem tools for automated interference detection using outlier detection. If you're interested, I can give you the reference for that later. But
one last thing.
yes, Brendan has one more.
Mike was saying all the transitions measured. So that
Right.
doesn't mean go out and measure every transition, because there are transitions that will have extremely low selectivity, like Y1, Y2, like, so yeah, 3 to 5 transitions, but I guess I'm contradicting Sue on saying take the most intense one. I'm saying if you measured a set, then we have no evidence that I mean, statistically the evidence seems to point to some.
Yeah. So for triple quad, 3 to 5 sounds like an okay recommendation. For DIA, use as many are selective and interference free for quantification. But I mean, I guess in summary, I would like to say if you are truly interested in the best quant possible, DIA is not your choice. You should definitely be going for an SRM or a PRM method, a targeted method. If you're truly, your goal is the best quant that I could quant and anybody's ever quanted. If you're thinking more about quantifying on like a profiling way, maybe you're interested in putting together like genomic signatures, fingerprint, stuff like that, molecular fingerprinting type things, DIA might be a good choice because you can measure more, but then you would use DIA to generate hypotheses to go on and create a targeted method to then quant. We went over this, and I just wanted to say that the comprehensiveness of DIA is really in the quantitative world where I think it shines. So in yeast research, one of the gold standard data sets for quantification of yeast proteins is this Gamogami paper from a little over a decade ago, and it's the gold standard because no one ever wants to repeat this. So they individually tagged all of the ORFs in the yeast genome, and the yeast genome has 5,000 to 6,000 genes. Not all are expressed under normal conditions. So we estimate there's probably about 4,500 proteins that are normally expressed in a non-mutant strain. They tagged every ORF, and then they did quantitative Western blots for each of those proteins and quantified these 4,500 proteins. There have been mass spec approaches to doing this. One of the best, I think, mass spec approaches so far was out of the UK, Lawless 2016. They used SRM and recombinant standard proteins called qconcat proteins. And they did 92 hours worth of targeted SRM assays to measure about 1,200 proteins in yeast by mass spectrometry. So my latest iteration of trying this with DIA, with a 2-hour DIA gradient, I'm getting about comparable protein quant as the Lawless SRM method. So their quantitation, however, remember DIA is a compromise, their quantitation might be a little bit better than mine. That's still something we're researching is how comparable is the quantitative results. But this breaks down into different proteins being detected between these methods. So DIA is not going to be an exact replica of what you would see with an SRM method, right? Different instrument for DIA than for SRM. But I think one of the cool things is out of the proteins that both the DIA and the SRM method are detecting and quantifying, the DIA method is able to do the quant on many more peptides per protein than the SRM is. Because SRM, they're just targeting 2 peptides per protein, 2 unique peptides per protein. With DIA, it's every peptide. So on average, each protein is going to have more peptides that are being used to quantify that protein. So that was just extremely quick DIA. If you're staying for the capstone next week, I believe one of the speakers will be talking about DIA, and now you will be at least armed with some knowledge of it. But I wanted to move on to talk about a validation recommendation that came from the CPTAC working group. And Sue will be walking through this case study example of how CPTAC did their calibration and validation for a peptide assay. But I just wanted to show you what the CPTAC assay portal even is. So Sue on Monday morning was talking about these online repositories where you can start looking at data-driven hypothesis generation. So did anybody ever see this peptide before? I'm looking for a protein. Has anyone seen any peptides from my pet protein before? Was it quantitative? Was it not so great? So CPTAC Assay Portal is one place where you can ask those questions, and they have extremely rigorous calibration and validation experiments associated with all of the targets in there. There is kind of a stamp of approval that you can win from the CPTAC group for characterizing your assay. Assay characterization, assay validation might be used a little bit interchangeably, so if you're confused, swap one for the other. Where there's a whole set of 5 experiments that are broken into kind of 2 tiers. So there's like this low tier where you just did experiments 1 and 2. And if you go on for brownie points to 3, 4, and 5, you get like the gold star sticker for your lab. And we'll be going into more detail about each of these experiments, but these are kind of the standards for getting a peptide target added to the CPTAC assay portal. If you wanted to get into the real nitty-gritty, if what I present to you later today is not enough detail and this is just super exciting to you for some reason, like Sue, you can get all of the dirt on CPTAC assay validation if you go to the CPTAC website and then click About. And then under About, click Resources. You'll get navigated to this page where on the right-hand side, what I've boxed in red, has a guidance document, which is an extremely detailed document about each of these experiments. And then they even have a little schematic representation, which is the image I just had on my slide. So we're just about ready to turn over to Sue for more on the CPTAC assay validations, but any questions? That was a lot of information that I just threw at you. No questions so far? Gonna wait and see if your question gets answered later. All right, I will give it over to Sue.